So it is an honor for me to introduce our next keynote speaker, who's the speaker of the day, Dr. Susan McCouch. Dr. McCouch is the Barbara McClintock Professor of Plant Breeding and Genetics in the School of Integrated Plant Sciences at Cornell University. She has received her PhD from Cornell in 1990 and spent 5 years in International Rice Research Institute in the Philippines before joining Cornell faculty in 1995. She is well known for her pioneering studies in molecular mapping in rice and the development of genomic-based platforms to explore the extent and distribution and phenotypic consequences of genetic variation in Oryza. Researchers throughout the world utilize the molecular markers, genotyping platforms, analyses protocols, and germplasm resources developed in her lab for genetic research and breeding applications. She has trained scores of young scientists who contribute to rice research nationally and internationally. She also has a prolific publication record. She is the recipient of numerous teaching and faculty awards, is an elected fellow of AAAS. She has served as a scientific advisor on the Global Rice Science Partnership, the NSF Advisory Committee for International Science and Engineering, and as a chair of the AAAS section on agriculture and natural resources. The title of her presentation today is Linking Genome-Wide Association Studies and Genomic Studies to Better Utilize Natural Variation in Rice. Please join me in welcoming Dr. McCouch.
Thank you very much for that introduction. I think I'm allowed to stand out here, and since I like to waltz around, I'll do that. It's a great pleasure to be at Texas A&M, and I must say, had I had a career at Texas A&M, I would have had an advantage trying to work on rice and rice genomics because you at least can grow rice in Texas. We don't grow rice other than under glass in Cornell or up in Ithaca. Anyway, I'll launch into this so that I try not to go over time. I'm going to talk to you today. There are 3 major areas of the talk. The first is going to be on a little bit more on the background, a little bit linking to some of the things that Paul and Heather have talked about with respect to ancestors. And the second on genome-wide association, and then I hope to get to the third part, showing how we're starting to develop models for genomic prediction that take into account the major effects that we see in rice and discover through genome-wide association. So bear with me. It's a broad arc. So rice really represents a mosaic of diversity, and when you think of it at a landscape level, it's quite significant, and the way it's partitioned is very interesting. Keep in mind that despite the way we raise rice in the United States, most rice is planted on farms that are less than a hectare in size. 92% of rice consumed on the planet today is produced and consumed within the country of origin. It never hits an international market. So unlike most of the other major grains, rice really is not such a commodity, and it's very tailored to the local environment and the local preferences of the different cultures that consume it. That leads to the last point, which is that consumer quality is diverse and inelastic. If you have a local crop, it's going to cater to a local cuisine and local preferences, and this is probably the largest challenge to the rice community in terms of increasing yield. It's very easy to increase yield if you feed your crop to an animal. It's much harder to increase yield if what you have to do is also tailor quality to a particular consumer that has a very ancient and very strong preference for a certain type of rice. Many of you in this audience, I look around, you're shaking your heads, you all know, you come from different parts of the world and probably prefer different rices. So my lab undertook a kind of global survey to start with. We asked 3 questions. How much genetic variation is there in Oryza sativa? How is it partitioned and where is it found? The second, how can we use that diversity? See if I can work this. Yes, to identify genotype-phenotype associations for traits of interest. That's the reductive science that genome-wide association, very much like linkage mapping, is all about. And the third is more synthetic. How can we efficiently select on favorable alleles at thousands of loci, both of major and of minor effect across the genome, to increase the overall rate of genetic gain, in other words, to put alleles back together. So that's sort of the way in which this talk is going to progress as well. So how is diversity organized in Oryza sativa? I'll whip through this because many of you may already know this, but I just want to remind you the basis upon which the structure of the rest of the talk will proceed. We have two major varietal groups which we recognize still as the indica and the japonica, and by the way, we're moving into the concept that maybe we actually have three. But I'll leave it at two for now, just so that's not the point of the talk. There are 5 major genetic subpopulations that fall into those two groups. The japonica, which consists of the tropical japonica, which is what's grown widely in the United States, the temperate japonica, which we grow in California and obviously is temperate, so in far south is Chile and as far north as Japan or Korea and parts of China. And then the aromatic or basmati group, what we call the group 5 rices, which are also very northern and are part of the japonica group, which you see here in this PCA. And the indica group that consists of 2 much more divergent types, the Ausch and the indica. And as you see from just the distance between them in this principal components, these are much more evolutionarily distinct than the temperate and the tropical. We believe that the temperate is simply a derivative of the tropical. The basmati or aromatic looks like it's actually kind of an interesting origin. It has a combination of admixture between temperate japonica, Ausch, and indica, and it sits right in the middle of these groups, but its chloroplast is clearly japonica, so its maternal origin is clearly coming from the temperate japonica group. When you look at this, how this distribution looks across the globe, I'm just going to show you briefly. We have from the diversity that we've sampled and that consists of our diversity panels, the center of diversity is clearly in Asia. Outside of Asia, what you see is in West Africa, parts of East Africa, North and South America, predominantly the blue color. The blue color throughout this talk will be conditioned on the dark blue being the temperate, the light blue being the tropical japonica. So the blue is the japonica side of the equation. The red and the yellow are the Ausch and the indica, with the basmati groups hovering up here in a very distinct region of the world. I will just point out that while indica is grown in South America, it is predominant. It is the predominant rice that's grown throughout most of the rice-growing world. When we look carefully at the region that constitutes the Vavilov centers of diversity, they've long been recognized, and there is a long-standing, let's say, feud about whether there's 1 or 2 domestication centers for rice. And rather than enter into that feud, I'll just say that the distribution of diversity across the region can be sort of understood a little bit along 2 different axes. One is the north-south, with the temperate germplasm in the north, the tropical blue in the south, and the basmati, of course, is slightly more northern, so it has that tropical ancestry to it. We also have an east-west axis. To the east, you see that's predominantly, in the southern part, is predominantly indica. Moving farther west, you see the Ausch's. This explains why in Chinese we have only indica and japonica. They really don't have the Ausch. They never recognized the Ausch, and for a long time early on in our work, we would be turned down by Chinese reviewers because they said the Ausch didn't exist. The fact is it exists outside of the territory that is really known and has been long acknowledged as one of the centers of diversity. So there's clearly another center of diversity that hovers in the Bangladeshi area where the Ganges River comes down. And I won't talk too much about all of that except to say that right here in the center, you see the pie is most diverse. So right in the center of Southeast Asia is where actually, both today and we believe ancestrally, the maximum amount of genetic variation for Oryza sativa continues and historically existed. It is very different in the regions that extend down into the outer regions of where rice is grown. You do have very novel genetic variation, but the maximum amount is found right in the center. And I'm going to tell you a little bit about some work we've done over the last year that I think explains why that is. Before I go there, think about for a minute this connection, this integration between geography and genetic diversity in light of all we've heard about this morning. First of all, the ancestor of rice, which of course you see here, has about twice as much genetic variation as the indica or the Ausch groups. The indica and Ausch, each one alone, has about twice as much as any of the japonica groups. And the basmati and the tropical japonica have about twice as much as the temperate japonica. Given the temperate is a derived group, it's a bottleneck that went very far north. That doesn't surprise anybody, I'm sure, in this audience. But it's noteworthy that our reference genome is in the temperate japonica group. So the cultivar Nippon Bare, which is a Japanese cultivar, is our reference genome. And so despite the fact that it couldn't be farther from the center of what is currently Oryza sativa diversity, we are all aligning, most of us still, to this reference genome, which is from the temperate group. What does this mean in terms of genome-wide association mapping and in terms of breeding? It has something to do, we're going to talk a little bit more about what this diversity means, but what it means for genome-wide association mapping is in terms of LD. So the linkage disequilibrium that we see in Japonica, in part because of its low diversity, is about 10 times higher. In other words, our linkage blocks are much bigger in the Japonica group on average than they are in the indica or Ausch or in the wilds. And so you can see that there is a progressive diminution, if you will, a decay of linkage disequilibrium, which means that you have much higher resolution if you're doing GWAS, genome-wide association studies, in indica or in Ausch than you would in Japonica. But it doesn't mean that you can do them in indica and learn much about Japonica, as I will show you in a moment. So it's quite interesting that we have this range, and so it's a sort of comparative opportunity for us to think about both how the degree of variation affects what we're trying to do with genome-wide association, but also whether or not we're actually mapping the same variation in these two very different groups. I'll mention that when you look at gene space, when we do de novo sequencing, instead of aligning to the Nippon-Bare genome, if we do de novo sequencing, and this is a paper we published in 2014, this is just de novo of an indica, an Ausch, and the reference genome, the temperate Japonica, we see that the indica is as different from Ausch as either one is from Nippon-Bare. So this is part of the sort of coming story that's breaking now about the possibility that we actually have three very, very different ancestral groups, each of which gave rise to domesticates. But the discussion about when and how all of that occurred, I'm not going to enter into today. Okay. So in order to look at where the variation comes from and whether or not it is likely to be similar in these groups, and in other words, whether they're derived from each other or whether they're derived independently, is important because breeders want to know where to look for novel genetic variation. They really need to know what is ancestrally most related to what they work with. So here I'm just showing you the features that are considered to be distinguishing between the two primary closest ancestors to Oryza sativa. The Oryza rufipogon, which is considered to be a perennial species, and the Oryza nivara, which is considered to be an annual species. These are still not very clearly differentiated in many studies. In fact, they're very confused. But here are the features that are supposed to differentiate these species. The habitat for the perennials is deep water. We believe that climate change 10, 12, maybe 14,000 years ago, where we started to get a monsoonal rain pattern, where we had a wet season and a dry season, particularly in South Asia, is what drove annuality. So annuality itself is a derivative, we believe, of the ancient perennial ancestor, which arose in an aquatic environment. I won't go on about all these because I'm not going to pursue that here. I just want you to keep in mind that there's still discussion about whether or not, for instance, one is derived from the other, and discussion about whether or not they interbreed. We published in just last year in the journal Rice a study on 286 wild accessions from the IRRI gene bank that were selected to represent a broad sweep across Asia, 15 different countries in Asia. And these wild species were a mixture of Oryza rufipogon and Oryza nivara, and they're just named by the person who collects them. They were not based, that was just simply based on climate, on the ecological and the morphological observations of the collectors. What we found when we put together, this is an unrooted tree based on GBS SNPs, so we had about 130,000 GBS SNPs in this study. What we see is that there are some very interesting, so this is the unrooted tree, and this, by the way, is the structure diagram. Many of you will be familiar to this. What I want to show you is that there is, in fact, a group represented here by the green and the orange on this end, close to the indica and the Ausch side of the Oryza sativa tree. And there's another group represented by the blue and the purple over here on the Japonica side of the tree. And when we match that up with the way things are named in the IRRI gene bank, what you can see is that the rufipogon, which is this black line, and the Navarro, which is this blue line, correlate very clearly with these two clusters in the phylogenetic tree for Oryza. We call this the Oryza rufipogon species complex. There is no very clear distinction. As you can see that there are some things that we would classify on this side of this that are classified as Navarra, and there are a few of these that are classified as rufipogon. Either they're admixtures or they're simply misclassifications, and we're not making a doctrinaire decision about that, but what we're trying to show you is that there are in fact still two fundamental groups, even in the wild ancestors, at least those that exist today, that they cluster very differently. But what's interesting about this tree is, of course, that there's this triangle in between. And the interesting part about that triangle in between is that we're hypothesizing that this is in fact the ancient population of perennial ancestors, because you see this black is the original, well maybe you don't, but this is everything is derived from this when you make your structure. This is the thing that sits at the heart of the wild ancestral group. And it includes both annual and perennial classifications. And so we're hypothesizing that actually annuality may have arisen several times for reasons of climate change in the past, and that it acts on a perennial ancestor when that perennial ancestor is removed from a perennially deep water situation and has to undergo some sort of seed propagation in order to survive a dry season. You can see where I'm going with this in terms of some of the challenges we're facing today. I'll just show you the correlation back with geography here to again show you that we have the indica side of the tree, indica and Ausch side of the tree in Western Asia, the Japonica side of the tree in Eastern Asia, and right in the middle sits what we would call today the major group of intermixture and what we call today indica rice. The explanation we give in our paper is that indica is both annual and perennial. That helps explain the maximum diversity that we see in the indica and the Ausch subpopulations, because they are in fact derivatives of, and they have experienced a lot of climate variation. They are adaptations to, and they're grown throughout, and they're admixed populations in the sense that they have absorbed genes from both gene pools. I won't pause on this because I don't think we're going to have time, but that's where the greatest diversity is, both genetically and geographically. So we put together a genome-wide association panel, and we wanted to take into account as much of the genetic variation that we knew existed as possible. The RDP1 was constructed in collaboration with our USDA colleagues, and it was about 400 Oryza sativa and 100 Oryza rufipogon species complex individuals. And then the Rice Diversity Panel 2, which is a much larger panel that was put together in collaboration with our colleagues at IRRI, consists of 1,200 Oryza sativas. And one of the major differences is that here we have most of the tropical germplasm that grows very well in the Philippines. Over here we have a relative paucity of the indica and the Ausch because it doesn't grow as well in the United States. But together we have 1,600 homozygous. They've been purified for several generations, Oryza sativa accessions, and my lab put the 700,000 SNPs on them. These are 700,000 very high-quality SNPs, and that's publicly available now. Now, as you may know, IRRI's also sequenced 3,000 genomes. Those are a completely independent set, but the good news is we've been able to impute from the 700,000 using the 300,000, we can impute up to full sequence. So the rice community is in clover, if you pardon my saying so. We can actually think about having 3,000 plus 1,600 sequenced genomes. And many of them now have been purified and are publicly available. That is a very big resource, but it requires now that people get to work on the phenotyping side. Before I just go on to talk briefly about the phenotyping, I want to say when you look at how these things sort out, this is a de novo assortment based on the LD that we see. We see the temperate japonica, the tropical japonica, aromatic, indica, Ausch, and rufipogon. The take-home from this slide is simply that there is no such thing as a pure indica or a pure japonica or a pure rufipogon. Everything is admixed in rice. Sometimes it's misclassified. Clearly, this Italian tropical japonica is a temperate japonica, et cetera, et cetera. I could go on and on about it. I only show you this because I'm going to come back and use it again. But the point is, all these little things in between are introgressions from one subpopulation to another. The subpopulations have maintained identity over all these years, and they're clearly distinguishable. And that all in and of itself is quite remarkable. So we have 5 different subpopulations with integrity, with clear haplotypes, and we can then look at how things are introgressed into the wild or from the wild, if you will. Okay, phenotypic evaluation is a very, very important part of understanding our germplasm. We have, in collaboration with our USDA colleagues and with IRRI, we've launched an international phenotyping consortium, if you will. People are free to order those lines from IRRI or from the USDA, and they can download the SNPs from our website, and they can get on with their phenotyping and do GWAS or anything else they want to do with these lines. I will put one caveat out there. We have discussed it briefly in this symposium, and that is, of course, you have to sign the SMTA now that the United States is a signatory to the treaty, and of course, IRRI, a lot of these things are maintained in the IRRI Gene Bank. So for those of you who are happy with signing the SMTA, that's good news. For those of you who are uncomfortable with it, that's not great news for anyone. Okay. What are we finding when we do genome-wide association? I'll just show you some take-home messages so that you can think about what it means to do genome-wide association when you have 5 subpopulations. One is, this is when we put all 5 subpopulations together. We're looking at grain length. And what I'm going to show you in the next minute or so is that using genome-wide association and dissecting these into the different subpopulations, we can find subpopulation-specific variation, which tells you that if you're working in indica, even though you have maximum genetic variation in indica, there's a lot of variation that japonica has that you don't have. So although you have a lot, you don't have it all. So even the subpopulations within this one cultivated species have a lot to offer to each other. So this is just a very brief take-home. Here is all for grain length. You see 2 very big peaks and a couple of smaller peaks. If you just look at indica, you see the same big peaks, all right, for determining grain length. If you look at Ausch, this one is completely missing. This one is much smaller, all right, but you see other peaks that are peaking up in Ausch. Those are the 2 indicas. When you look at tropical japonica, this peak retains its significance. This one is much lower. And when you go to tropical japonica, this one is practically gone, and this one is actually a different peak that's just to the side of it. So different subpopulations just for grain length are, and I use this because you all know, if you eat sushi rice, you're always eating small grains. If you're eating indica rice, you're probably thinking of long, thin grains. And so we can see that there is different reasons. There are different underlying genetic loci that have been selected on for this one trait. And the other important thing is that, so sometimes in all, you see things that are present in some but not all of the subpopulations. Those are the big peaks. Sometimes you see something that's present in all, but it only maps back to one of the subpopulations. And then, okay, in this case, it maps only to the temperate japonica. And then there are subpopulation-specific peaks that you don't see because the alleles are not at high enough frequency when you put them all together, but they are at high enough frequency to obtain significance in one specific subpopulation. So you're really mapping on what would be almost different species here. You can combine them, or you can look at them separately, and you can learn a great deal about where these alleles come from and which allele you might want to use if you're a breeder. And I'm going to show you a few examples. But let me just say, this is one example of a Ausch-specific allele that is kind of the first time anyone had ever diagnosed it. It came out of this particular GWAS study. So the gene that controls the long grain has been cloned, and the functional SNP happened to be included on the SNP chip, on the 700,000 SNP chip. So it was very diagnosable in our GWAS for either all, but it's completely missing in the Ausch subpopulation. However, in Ausch, there's a second SNP that's very low and very insignificant when you look at all, but it is highly significant in the Ausch subpopulation. And I'm just going to show you the haplotypes. So there are 3 basic haplotypes here. We see the japonica haplotype, which is all blue. These that have yellow are actually recombinants, so they have an indica haplotype up to here, and from here it's completely japonica. They carry the functional mutation for long grain. Long grain that we think of as an indica trait actually originated in a tropical japonica and was introgressed into indica. And every indica that carries that particular allele for long grain, which is a majority of indicas, has the blue haplotype across the entire region. So we know that indica got its long grain. Indica is originally a very small grained thing, but it intercrosses very well with tropical japonica, and there's the tropical japonica haplotype right across there. In Ausch, and here I'm showing you Ausch, there's a unique mutation very near to the gene that confers the long grain in indica and in tropical japonica that is not seen in any other subpopulation, at least not in our survey. That particular allele that's present in Ausch, not at high frequency, but in a significant number of lines, makes grains long. So this is the Ausch allele. This is the wild type allele in the Ausch, which is a short grain, and the derived allele, which is different than the long grain allele that we see in indica and japonica. It's an Ausch-specific allele that makes the grain long. And just to mention that previously we had reported there are already known to be 5 different alleles in this same gene, some of which make the wild type longer and some of which make the wild type shorter. So rice is full of variation for grain size, which has been one of the targets of selection. But there are lots and lots of these things, and I think we heard about it earlier in other crops as well. We've got lots of alleles segregating, and in an inbreeding species like rice, it's classic that because we have very isolated populations, and as I said at the beginning, they're selecting for local cuisines and they're working in inbreeding crops, we have many local varieties that are maintained as isolated populations. And if we go in and look at those populations genetically, we find lots of novelty, lots and lots of novelty. And I don't want to spend too long on this, but I think it's an interesting example and it follows very well from some of the work we just heard from Heather and from Paul. Here is the allele that confers aroma in rice. It's been cloned. It's got 15 exons. It's called the bad H2 allele. And we undertook a diversity survey to look at variation in this one gene in rices that were collected from the IRRI gene bank that had come from different parts of Asia. We found multiple, in this case we diagnosed 10 novel alleles, all of which led to, here is the amount of 2AP, which is the aroma compound that you sense in the human nose, showing that these alleles actually correlate with an increase in aroma in rice. They come from different parts of Asia. Each one can be mapped back to the specific area in which that landrace was collected. And what we're showing is that despite the incredible diversity of alleles for aroma, there is one which is predominant, and that one comes from the basmati origin. It's found in 65% of aromatic accessions in the gene bank, but 95% of accessions on the market today all carry a single aroma allele. If you go to Southeast Asia and you order rice in any village in Laos or Cambodia, you're probably eating a different aroma allele, okay? So enjoy. Now, to show you what the evidence looks like on this kind of panel, you've seen it before, I'm just going to show you things that we can start to diagnose. This is the SD1 gene that came in during the Green Revolution. You see it's an indica gene because it's red. It came into the tropical japonica. This shows you something about the kind of thing we can see. When they're recent introgressions, the introgression itself is very big. When they're ancient introgressions, the introgression is very small. What I'm going to show you is evidence for introgression of the aroma allele at the fragrance locus on chromosome 9 and on chromosome 8, and I'm going to show you what that looks like close up in an indica, which happens to be a fragrant variety, and you all may know it by name. It's called Jasmine 85, and if you go to the market, it's one of the supreme quality rices that you pay for. Very nice aromatic soft white grain. Jasmine 85 carries, I don't know if you can see it in this light, a little purple introgression right on that black line is the fragrance gene. Can you see it or not? I don't know in this light. So Jasmine 85, which is a Thai jasmine, which has been patented, and the Thais are very proud of this rice, happens to carry from a very ancient introgression event a gene which obviously originated in a basmati ancestor. We can show the same thing for a tropical japonica called Azucena, which is a popular aromatic variety in the Philippines. And I'll just show you that again. You can see the introgression right at the fragrance gene in Azucena coming from, that's the BADH1 introgression. In other words, we know exactly which allele it is. I'm not going to go on and on about all the things that we've discovered and we're discovering with our genome-wide association. I just want to show you an example from some work that's been done on roots, where we did image analysis on 10-day-old seedlings in gel and gum. There is nothing more frustrating to a breeder than to hear a molecular geneticist from Cornell come and talk about doing work like this in gel and gum in a test tube and then pretend to be a breeder. That is an assault on any breeder's normal logic, okay? So that's why I want to tell you this story. There are reasons to believe that one day basic biology was going to have something to offer to the applied breeding community, and I think that day is almost upon us. So here's my example. We did 3D root system architecture. Oh, and by the way, I do have to just say, we sequenced 40 images per plant on a rotating, on a rotating system in water to be able to capture these images. You can actually see the root system. And then they were imaged over time, different days, and then we used a software package which we created with this wonderful graduate student who was very gifted in software engineering, and he put this together so that we could concatenate images and reconstruct a 3D image of the root system. So what I'm going to show you is, quickly, we did both genome-wide association, which are the red dots. You see they're much higher resolution than the QTL analysis, where we did biparental QTL analyses. And I'm showing you that in some cases the QTL and the GWAS confer. I mean, they're in similar regions, okay, but many times they're in different regions because one is a biparental, the other is a GWAS on this large panel. What we diagnosed is many hundreds of QTLs. We had this image analysis, and you can compute anything you want. But it comes down to, sorry, I guess my, there it is. This is the genome-wide association analysis. You see on chromosome 4, here's a major peak. This peak is actually a GWAS peak that does not coincide with a QTL on our biparental. And what you see is that the concatenated images are going to show you something about the resolution that we get in 10-day-old seedlings. First of all, this is the region we're looking at. We have SNPs that are 66 kb away, 33 kb away, or at the peak SNP. The peak SNP was that SNP in the diagram I showed you. 66 kb away, there is no difference in root length in our little gel and gum system. This consists of a concatenated version of a 3D image of 39 different accessions from the GWAS panel that carried the A allele at that SNP, and 118 that carried the B allele at that SNP in the indica subpopulation only. And what I'm going to show you is as you move in, when you get to the peak SNP, you can see that there is a significant difference in root depth, in gel and gum, in a test tube, at 10 days. I know, I feel very small when I have to tell this story. But the good thing was that it was very repeatable and very predictable. And so here we are saying that in indica, we have a SNP that can predict longer or deeper roots than at that alternate SNP at this location in the genome. And that's a pretty exciting thing, but does it have any impact on the field? So what I want to show you is first, before I show you the rest, is that when you look at the different subpopulations and you think about this particular SNP, we can show you the lines that carry the two alternate alleles. And what we show you here is, see the blue dots? I don't know how clearly you can see it. In the indica subpopulation, the blue dots are the allele for are a japonica allele, it's coming from this, that at that SNP is very frequent in japonica, and it is clearly, those are the lines that have the lowest deep rooting depth. And here are the indicas, and you can see the dots up here in the tropical japonica, just based on the introgression analysis that we're able to do in existing material, without any other further genetics. You could go in and choose tropical japonicas that carry that particular allele. And in fact, if a tropical japonica did not carry that allele, but already had deep roots, what we are predicting is that if you add that allele, you will deepen the roots of that tropical japonica. Now, meanwhile, while we were doing this work at Cornell, unbeknownst to us in Japan, they were carrying on field experiments. And at PAG 2 or 3 years ago, I got up and gave this talk about our GWAS, and following me, Dr. Uga got up and gave this talk about 15 years of field research, where they had gone to the trouble of diagnosing rooting depth by digging up plants and using this colander where they actually planted the plant into this colander and they could measure the rooting depth and the rooting angle. It's very beautiful work, and they showed that, in fact, there was a difference in rooting depth based on root angle, and they cloned this gene. And this gene turns out to be one of the very genes that we diagnose in our little system at 10 days old in our gel and gum system. So, for one thing, root system architecture is becoming a really interesting area for people to work. But when I started this work, which was in 1995 at IRRI, we started our first, and we reported a QTL paper on rooting depth in hydroponics and in field. And my supervisor at IRRI told me this was a figment of my imagination. So I feel that we're coming full circle. And if we work together, there are amazing things that we can learn and how we can bring these things together. And I don't have much time, so I don't want to pause too long on Dr. Uga's work. But this young man has done amazing things with this system, and he started with it in the field, and they're able to clone this gene that shows that this simple angle makes a difference. And here's his data, not my data, here's his data. He did marker-assisted selection. He introgressed the allele for deep rooting into the IR64 background, which has shallow rooting, which has been bred for irrigated systems. And he showed that under no drought, the IR64 and the near isogenic line carrying the deep-rooted allele have no difference in yield. Under moderate drought, the IR64 loses yield fast, and the NIL is significantly unperturbed by moderate drought. Under severe drought, IR64 dies, and the NIL is still able to yield. So that one change, this is a single base pair deletion in exon 4 of this gene, has a significant effect on a field trait that we all know and think is important. And we are able to detect it in gel and gum at 10 days old because genetics is a very powerful thing. So look up Dr. Uga's work. He's cloning more of these now, and the good part of the story is he came to Cornell to learn how to do this work in gel and gum because he thought, wow, I spent 15 years in the field. And these people spent, one PhD. And now we're colleagues and we're collaborating. Okay, now I'm going to move to the last part of my talk. I'm going to talk about marker-assisted selection, which you can do when you have a single base pair change and you can target it and it has a big effect and it's going to matter. You can do marker-assisted selection for disease resistance, for many of these traits where you have a major effect allele. But one of the problems we have in the rice community is that by and large rice breeders have not been trained as quantitative geneticists. They've not been trained like the maize breeder. They've not been trained like the sorghum breeder. They're not thinking quantitatively. They're thinking, because we have this beautiful system where we have genes of big effect, they've been thinking, as I was thinking for a long, long time, about big effect genes and marker-assisted selection. And the rice community is really good at that. They're really good at that. And it's making a difference on millions of hectares of land. So it's nothing to be ashamed of. But it's time for us to start to think quantitatively. And so we really are starting to look at how we can actually start to move our genetic gain forward at the quantitative level as well as at the qualitative level. We have lots of quantitatively inherited traits that haven't been moving forward as fast as we need them to move in order to meet, obviously, the objectives of the world food supply. So we started looking at this in collaboration with IRRI and in collaboration with partners in Uruguay with their national program. And that's been interesting because you have one case in the tropics and one in the subtropics, and as you all know, Uruguayan rice breeding and rice production is very similar to what we find in the southern United States. So it's kind of an interesting proxy for what we're starting to do here in the US as well. At IRRI, we had 331 advanced indica inbred lines. That was just part of what was in the breeding program at the time we started this collaboration. And we had data from their replicated yield trials from 2009 to 2012. we put 73,000 SNPs and measured 11 traits in a collaboration with a wonderful PhD student from Bangladesh on this IRRI irrigated breeding trial. In India, we had a very similar number of lines, so we have a pretty good, pretty parallel situation. We're working in the tropical japonicas. We have replicated yield from 2010 to 2012, and we're trying to move that up to 2014 now. And we have about 40,000 SNPs. Of course, this is a much narrower gene pool, as I mentioned to you. So although the GWAS was done very similarly, there's simply less polymorphism in the tropical japonica. What I'm going to report on is the work that we've done in collaboration with IRRI, where we looked at yield, plant height, and flowering time, and that's just the summary I'm going to provide for you right now. The heritability of these things is not very high, but you can see that flowering time is a bit higher than either plant height or yield in the IRRI situation. And what I'm going to tell you about is that the accuracy of our predictions are mimicking pretty much at this point in an unbiased way. We're just able to do pretty much what you could do with your phenotyping, which means that, okay, that one generation of genomic selection is about as good as a whole nother year of phenotyping. So it's not better, but it's not worse. So if that's going to help you accelerate your breeding, just the first pass means we were doing okay. We looked at lots of different models, and for those of you who are doing GS, you may be interested in this. For those of you who are not, I will just mention that when the trait is very quantitative, it turns out that certain models predict better. When the trait has a lot of major effect alleles, then other sorts of models affect accuracy. So depending on the trait, you can optimize your model to predict your trait. If you know beforehand that it's highly quantitative, that will tell you one thing. If you don't know, one thing you can do is just do GWAS, run your GWAS model on the phenotype and genotype material that you have, and determine whether you should, which model. You can predict that fairly well. The other thing that we're doing is we're looking at how we can use the IRRI model, which is a very, very efficient model, and improve the accuracy of that model by incorporating essentially the major effect, putting weights on the SNPs that tag the major effect genes. And what I'm just going to summarize here is this paper that was published in 2016, showing that in fact when we look at flowering time, where we have one major QTL in the IRRI breeding material that we were looking at, versus yield where we have one major QTL but only predicted 10% of the phenotypic variance, where the flowering time major QTL predicted 45% of the major phenotypic variance, or plant height where we have 4 different QTLs together predicting about 74% of the phenotypic variation. We have dry season and wet season. These are just showing prediction accuracies, and the take-home message here is that we can improve prediction accuracies if we add a GWAS weight, GWAS-based weight to the SNPs that are identifying these major QTLs. So we can improve our predictions for flowering by about 35% if we just put a weight on that QTL. We can improve yield by about 14% because, of course, the QTL doesn't predict as much. And we can predict plant height. We only added, actually, I think 2 or 3, not all 4. We can improve up to 20% our prediction accuracies. The point is that if you have a model that assumes that all SNPs should be weighted equally, and it turns out that in your material there are major effect loci, you do want to account for those major effect loci. And there are much more sophisticated models to do that now, so you're not going to go in and add these as covariates as we did. But I think the major take-home point is that if you know your organism and you know whether it's truly quantitative with lots of small effect alleles, you're going to expect a different model is going to be optimum than if you know you have a mixture of large and small. The other question that occurred to us was, can we just go into the literature and find out which SNPs we should weight? Because after all, people have been looking at flowering time and plant height and yield for a long time. Can't we just pull that out of the literature? We don't have to recalculate it every time. And it turns out that most of the time, see these blue, these are the de novo predictions about which allele and which SNP are significant compared to what's in the literature. So most of the time you do much better by using your own data, your own breeding material to predict which SNPs are segregating and explaining your trait than you do by looking at the historical information about those traits, because of course historically they're not using your breeding material. Sometimes you do significantly better, and sometimes you actually don't do much better, but you never do worse, okay? So we've determined that in fact, historical data, as useful as it is, doesn't tell you really what's segregating in your breeding material. You better do that and find out for yourself. And my final point is that this combination of genome-wide association and genomic selection is interesting because you start with the same matrix of data. And the matrix of data can be, you can run one model or the other. And if you like to do both, you can mix them in interesting ways to actually be able to pull out major effect alleles from unadapted germplasm, which you would have to say is exotic. But once you've got those new alleles introduced into your breeding material, your elite breeding material, and they become more frequent, then you can use a standard GS to incorporate them, and you can put different weights to carry or to discard certain alleles across your genome. So I think there will be interesting opportunities for people to go fishing in the gene banks and in the unadapted germplasm, bring in new alleles, and then use GS to expedite their delivery into new varieties. And I guess we're at the end of our time, so I'll just say, here's my wrap-up. GWAS is helping develop a genotype-phenotype roadmap to accelerate trait and gene discovery in rice. Genetic architecture of most complex traits in rice involves alleles of both large effect, which are amenable to GWAS, and genes of small effect, which are more amenable to genomic prediction or genomic selection. We have very different genes or alleles that are important in different subpopulations, in different wild and different cultivated populations. And so where you go to get what is exotic may change based on what you're actually trying to bring in. And finally, the use of de novo GWAS and GS, where you weight alleles of large effect, can enhance the accuracy of your genome-wide prediction and hopefully increase the rate of genetic gain. And with that, I hope there's time for at least one question. Thank you.
Well, thank you, Dr. McCouch. If we have any questions, we have volunteers on both ends of the room who will bring a microphone to you. Dr. Murray.
Thanks. I really appreciated your anticipation of the defense of plant biology for plant breeding. But I think there's two specific technical criticisms that I would still have on that issue, and I'm wondering if you've addressed them. The first would be context dependency and pervasive epistasis. So discovering these alleles in a model sequence genome and then putting them in a different background, the alleles tend to have different effects. And then the other one is the challenges with reverse genetics. You had a lot of candidates up there, and you confirmed one of them, but have the rest of them shown to be valuable?
Great questions. So the first one is genetic background effects, and I think we constantly address genetic background effects in rice. So most of our work is actually not trying to predict the effect of an allele when you move it across genetic background, but trying to use that mosaic to find where the allele is already in another genetic background and test that, the impact of that allele, so you use that as your parent if you're working in an alternate genetic background. And that's turned out to be surprisingly effective in rice. The other part of the work that I presented yesterday at the workshop is showing how much epistasis and transgressive variation we do find when we move things across genetic background. And I think that's a very important point and important take-home. You have to work within the gene pool that is adapted in your area. And it's not only epistasis. I think it's just even expression in response to environment that can be quite different. So I do take that very seriously, and I think that's an important thing for everyone to keep in mind. So the genetic background dependence of prediction. And yes, I bow to the importance of that. Your second point was, what was it, Seth? I'm sorry.
Genetics. You had a lot of candidates up there. You confirmed one out of many.
Oh, in the root analysis? Yeah, so this is the thing that obviously is taking the time. I think the very powerful part of having candidates is that you can then start linking those candidates. If you're doing, let's say you're doing transcriptomics and you're making, pathways, or you're looking at how all those QTLs are related to each other. And I think that I don't have time to talk about that here, but I think that the reality is, yes, you need to know a lot about the system that you're working in. It's not one locus, one gene. But I will say in defense of the one locus, one gene, that in rice we have enough historical examples of one locus, one gene that matter to farmers and that have actually huge impact on the farming community. Thank goodness we didn't say, oh, biology is too complicated for us to even bother with those. You start with what you have, you move forward. But I do think it's the era when we need to start thinking more about systems, more about these complex quantitative problems. And of course, high throughput phenotyping and big data is changing the game for everybody. And I guess maybe I just ask if you have a further response. I don't know if I've answered it. No?
Well, we have another question from the webinar.
All right, the question is, are all of your markers from GWAS? Are any of them from exome capture?
Mm-hmm.
And do you think we will have better results if we use exome capture since it will capture the genetic variations for GWAS?
So we haven't got anything from my group on exome capture yet. Doesn't mean that there wasn't a paper yesterday on that. So I don't know if there's a new paper in rice on exome capture. But I think the critical thing is that we have full genome sequence. So although we don't have the in-depth exome capture, which would be great, and as soon as somebody has the inclination, we hope that they will do that. But we do have pretty good information now on genome sequence. So we can go back into our fully sequenced genomes and look for sequence across any candidate genes we're interested in. How perfect is that sequence? How good are our imputations is another question. So I welcome the layers and layers of improvement that come with time. We never have more. We never have enough. So, but we haven't done it yet.
Well, we still have time for maybe one more question.
Thanks, Susan. It was really interesting to hear how many genomes you guys now have in rice, but it's all based on the one reference genome. So I was wondering if you could talk about how that affects your analysis, if there's efforts to try and understand structural variation in rice, things like that.
It's actually not all based on one genome. So what has happened is that we have now maybe 7 PacBio de novo assemblies for sativa. And then we have the OMAP project for all of these wild ones. And people have started aligning to different genomes, but there's no pangenome yet. So it doesn't solve the problem to have other de novos unless you can compare. So I'm part of a collaboration with Cold Spring Harbor, and other members of the rice community are working with different people to try to actually sort out where the regions of structural variation are. And what I find, where we will come out with a paper very shortly, is that the structural variation is as abundant within as between individuals. For instance, 2 indicas are as different from each other as an indica might be from a japonica. The japonicas are less different from each other. But the variation that we see isn't enormous. It's more at the level of gene duplication locally, some transposon activity, but not anywhere near like you have in maize or even in wheat and barley. So rice is a very constrained genome. Its gene order is quite conserved, and the amount of difference between even an indica and a japonica when you look at whole genome, de novo, PacBio assemblies is not it's distributed with very small structural variants in lots of different places. And what I fear is that people are going to come up with mandates saying, these are the differences between indica and japonica based on one japonica and one indica. So it's very good that we have actually 7 or 8 right now of these, so people are starting, but it's going to delay it a little. There are lots of structural variants that are small, and then there are a few that are very large that are easy to discover and that make a difference. So we don't yet have, for instance, open chromatin maps in rice, or there's been one that's been started, but I think some of these things still need to come together in the rice world. Yeah, and I do think they'll matter, but we're not there yet. Yeah.
Well, I guess we're at the end of the time for our question and answer
Thank
session.
you.
I would like to thank Dr. McCouch once again on behalf of all of us. So I would like to remind all of you once again that we have our social media channels, and if you're posting online, to please tag us on all your posts. We are about to break our session for lunch. Before we do, I want to thank all our sponsors once again. So I would suggest that everybody